Welcome life science learners to another lesson. Trust that you guys are well and looking forward to our lesson today. In our lesson today, we're going to be focusing on revising a very important topic, DNA and the code of life. It's important that this concept is reviewed constantly. It often forms the basis for many other topics to come. So let's get straight into the revision lesson. Let's have an overview of the sections and then we'll apply some of our understanding. It's important that you, as you sit at home, are able to make notes. I often remind you to have a set of terms that are important. And so that's where we start in our lesson today. We're going to go through a list of terms, help us to refresh all of these terms so that when we get into applying these terms in questions, it becomes easier. So let's overview some terms and then we'll apply our knowledge in answering some questions. So it's important that we understand the overview of DNA. We're going to spend some time looking at the processes of protein synthesis and replication, and then we'll get on to discussing mutations and also then spend some time looking at some revision activities. As I mentioned, terminology is fundamental to performing well in life sciences. It's important that you draw up a list of these terms. As I have here, I've indicated some of the most important terms that frequently appear in this topic. So let's get through them, revise, recap, and I hope this tickles those memory cells so that these terms come back. So let's get into that. So guys, the concept of chromatin network, again, go back to the nucleus, and that's where we started off in the year when we spoke about DNA being the molecule of life, and essentially that's in the nucleus where all the chromatin network is found. We obviously spoke about chromosomes and how those chromosomes or the DNA arranges itself into these compact bodies called chromosomes, which are essentially made up of proteins, histone proteins, around which the DNA is wrapped. So we do have the histone proteins as a construction protein of the chromosome, and we find that the DNA is wrapped around that to form these complex structures called chromosomes. In this specific chromosomes, we mentioned the term chromatids, and essentially chromatids refer to those copies of DNA that have been produced after replication and then held together by the centromere. So it's important that you recollect the structure of the chromosome, that it's made up of chromatids and the centromere that holds the chromatids together. And that's important in the next section when you look at meiosis and the revision around that. The concept of a gene is very important because often that relates to genes and how we understand the concept of inheritance. So a gene is again a sequence of nucleotides that code for a characteristic, and we see the importance of that in terms of protein synthesis and how proteins are made. It's important that we understand what a gene is and what happens to the gene when a mutation takes place. So it's important to recognize what a gene is. A nucleotide again refers to the basic building blocks of nucleic acids. And remember, the two types of nucleic acids are DNA and RNA, and so we talk of them being made up of these simple building blocks called nucleotides. The nucleotides contain nitrogenous bases, and we refer to them as your ATGC base pairs, and so that's important when you're looking at adenine, thymine, guanine, and cytosine as being your DNA bases or your nitrogenous bases, as well as adenine, uracil, guanine, and cytosine for your RNA nucleotide bases. So that's a very quick recollection of the nitrogenous bases in both DNA and RNA. Again, a recap of what DNA is. You should know the structure of DNA. You should know the similarities between these two molecules in terms of the location, where they are, what are they made up of, and essentially the differences between them. And often we tend to revise that section towards the end where we talk about the differences between RNA and DNA, often in a table, and that's an important comparison to have. The significance of mitochondrial DNA in the mitochondria and its link to maternal ancestry, and that's important, and how we use that to determine relatedness along the maternal line, as well as we talk about the different types of RNA, where we've mentioned messenger RNA, transfer RNA, as well as ribosomal RNA, which we see all being important in the process of protein synthesis. Again, a quick overview is that the ribosomal RNA is our RNA, which is what the ribosome is, and that is an important part when we discuss protein synthesis. We mentioned the term complementary base pairing, and that again refers to how the bases connect on a double-stranded DNA. And again, the terms purine and pyrimidine need to come back because those are important in terms of identifying the different types of bases. And a very important concept, DNA replication, as an important process that we need to understand, as well as chromosome mapping in terms of the Human Genome Project. Okay, let's have a very quick overview of the process of DNA replication. It's important that we remember that DNA replication occurs in interphase, and it's a process in which DNA undergoes copying. And so it's important to remember these keywords where DNA unwinds and unzips, exposing the DNA templates. And these templates then become available so that new or free DNA nucleotides can base pair with these templates. We also recognize that the base pairing is facilitated with an enzyme, and that enzyme allows for base pairing to occur in a complementary nature, and we discuss all of that, and eventually we talk about how the base pairs link up together and how the DNA helix reforms. And this often comes in a question for about 4 to 7 marks where you describe the process of DNA replication. So you need to know what do you need, when it takes place, and how it takes place. So again, the enzymes, the free nucleotides, as well as the DNA templates, and it happens during interphase, and that's essentially the importance of that process, looking into mitosis and meiosis. It's important that the next concept is protein synthesis, and you review that concept well. And the best way to revise this section is actually to have a few diagrams that illustrate the two processes that are important in protein synthesis. And we'll have a quick overview of that: transcription and translation. And a reminder that transcript, again, is the formation of a message, and that is mRNA. And translation is changing that mRNA into a sequence of polypeptides. And we refer to those sequence of polypeptides being a chain of polypeptides, which will eventually combine with other polypeptides to make what we refer to as proteins. So that process, we'll very quickly overview that. You should know that there's a difference between where transcription happens, which happens in the nucleus, and the process of translation, which happens in the cytoplasm. What's important is that we recognize that transcription is when a gene produces a copy of itself which codes for information, and that copy is mRNA, and that mRNA then leaves the nucleus to move to the ribosome, where that ribosome is going to process the mRNA and read each codon and bring in specific tRNAs with their unique amino acids attached to them. The concept of codon and anticodon base pairing is important, and then being able to understand how amino acids are put together in that process. So guys, that's a very quick overview and a recap of some of the important concepts. Again, if you do not recollect these concepts, go and prepare some diagrams, look at your textbooks, and try and write down a few points around these 3 major processes: DNA replication, protein synthesis, where we talk about transcription, as well as translation. And so that's an overview. Let's have a short break, and then when we get back, we will apply some of this understanding to a few questions. Stretch around, get some water, and I'll see you in a bit.